Getting an innovator drug to market is a long and expensive proposition. Let's take a closer look. The whole process takes place in 4 stages, starting with the discovery phase with thousands of drug candidates. Getting an innovator drug to market is a long and expensive proposition. Let's take a closer look. The whole process takes place in 4 stages, starting with the discovery phase with thousands of drug candidates, taking it through preclinical testing or trials, clinical trials, and finally getting regulatory approval for a single drug product which goes into production. In the drug discovery stage, scientists identify a target, like genetic material, protein, or a cell, which may be leading to the condition. And they start with thousands of drug candidates to bond with the target. These could be small chemical molecules or large biomolecules or a biologic. Drug discovery requires heavy scientific lifting using techniques from structural biology to artificial intelligence. Of the thousands of candidates that start the discovery stage, only 100 or so make it to the next, preclinical stage. Preclinical testing involves non-human testing, like on rodents, guinea pigs, or monkeys. Preclinical testing is used to determine safety and efficacy standard for the drug candidate. Only a handful of candidates pass the preclinical stage to get to clinical testing. Next comes the longest and most expensive stage, clinical or human testing. This is done in 3 phases. Phase 1 of clinical testing is performed on a small group of healthy volunteers to establish safety and dosage criteria. Phase 2 is conducted on a larger sample of volunteers with a target indication to test for efficacy. And Phase 3 of clinical testing is done on a much larger sample and usually in many different locations, where the drug is compared to the current standard of care. Only one drug candidate makes it out of clinical testing to go into regulatory approval. Developing an innovator drug is a complicated process which can involve many different companies, from startups in Boston to contract research organizations in Hyderabad. It can take 10 years and over $1 billion to complete this. For this reason, innovator drug companies are usually chasing drugs that have the potential to become blockbusters. For much of modern medicine, pharmaceutical products have been based on small chemical molecules synthesized in the lab, like aspirin or acetylsalicylic acid. Each molecule of aspirin has just 21 atoms. Small molecules like aspirin easily gets into the bloodstream and penetrate cells. That's why it's used to relieve pain and bring down fever for millions of patients for decades. Because they are highly stable, small molecule drugs usually come in many different dosage forms, like tablets, capsules, and even topical forms that you can apply on your skin. They are easy to handle, and most of them can stay in your medicine cupboard for months or even years without spoiling. No wonder that small molecule drugs are so popular and make up over 90% of all drugs in circulation. But what they have in stability, small molecules lack in specificity, which means they work on the drug target, but they also affect a lot of harmless or even beneficial cells and body functions too. Think about chemotherapy drugs, designed to target fast-multiplying cancer cells. But chemo drugs can end up targeting other fast-multiplying cells, like white blood cells, reducing immunity to infections, and hair roots, causing hair loss. So scientists over the last few decades have been exploring beyond small molecules on how to make highly specific drugs. And they've been looking for inspiration to the body's own immune system and large biomolecules, especially protein antibodies that are produced by the immune system to target extremely specific conditions. This field, called immunotherapy, led to the birth of large biomolecule-based drugs. Compared to small molecule drugs with less than 100 atoms and molar weight less than a kilo, A typical biomolecule has over 3,000 atoms and can weigh 100 times as much. And monster large molecules like antibodies can have over 20,000 atoms and weigh a whopping 1 ton per mole. Because of their sheer size, large molecule drugs are not easily transported by the bloodstream, instead relying on the much slower lymphatic system of lymph nodes to get to various parts of the body. And they're highly unstable. Pretty much the only dosage form for large molecules is liquids for injection, which need to be produced, stored, and transported through highly specialized systems. But large molecule drugs are highly specific, binding with exactly and only the drug target, and having next to no side effects. For this reason, large molecule drugs based on immunotherapy are considered the future of pharmaceuticals, accounting for 7 out of 10 top-selling drugs worldwide in 2020. Here's a quick recap of the small and large molecule drugs. As the name suggests, the size of the molecules are vastly different. Small molecules have low specificity, causing side effects, whereas large molecules are very specific. Small molecules get absorbed and transported in the bloodstream, unlike large molecules, which have to rely on the lymphatic system. It's easy to produce small molecule drugs which come in many different dosage forms. Large molecule drugs, on the other hand, are unstable and usually come as only injections with special handling instructions. You can reverse engineer and make exact copies of small molecule drugs called generics. Large molecules, on the other hand, are complex and, like all biological products, can only be approximated to biosimilars, never exactly copied. Small molecule drugs dominate the market currently by volume, whereas large molecule drugs are low in volume but high in profitability, paving the way for the future of pharmaceuticals. The journey of an innovator drug starts with finding the right drug candidate to treat the disease or disorder. And the most popular method to find the right candidate is through a process called high throughput screening, or HTS. The aim of HTS is to find promising drug compounds, or hits, followed by refining them to get leading candidates through a process called hit to lead, or H2L. High throughput screening starts with identifying the drug's target, the protein, genetic matter, or whole cell that's responsible for a disease or disorder, and then screening thousands or even millions of possible compounds to see what binds with the target to influence its behavior. And how do you confirm binding between the target and compound? Through an assay, a chemical, biological, or physical method used to confirm binding. Here's an example of a biophysical assay. If you bombard a small, fast-moving chemical molecule with polarized light, it reflects off the molecule, giving out a unique reflected light spectrum, which can be recorded on a spectrometer. Once the same molecule has bonded with a larger biomolecule, it moves a lot slower, and the reflected polarized light spectrum is going to look very different, which confirms binding. So developing an insightful working assay is often the most crucial part of high throughput screening. At the core of high throughput screening is this: a simple piece of lab equipment called the assay plate, or microplate. Each opening or well in the microplate can hold very small amounts of the target and candidate, as little as a few nanoliters. So a researcher conducting HTS would fill the microplate wells with commercially available compound libraries, one compound in each well. Then she would add a small amount of the target biomolecule into each well, allow them to react, and run the assay to see which of the wells give you a hit, or proof of bonding. The output from an HTS run would look something like this. The blue wells indicate hits. Not all hits are great. Some are just accidents or results of lab errors, so they need to rerun the assay to see if the results can be duplicated, and then to see if the binding is selective. That is, the compound doesn't bond with unrelated biomolecules also. The hits that pass these further requirements are called leads, and only the most optimal leads make their way to the next stage of drug development, preclinical trials on animal models. Before a drug candidate can go from preclinical trials, where it's tested on lab animals, to clinical trials, where it's tested on humans, regulatory bodies require an early stage approval for the drug candidate. With the US FDA, this process is called the Investigational New Drug or IND application. IND applications require proof that the drug is safe and effective to use in human clinical trial volunteers. For this reason, IND applications have to be supported by IND enabling studies. IND enabling studies are broadly of two types. Pharmacological studies, which tries to answer the question, does the drug work? And toxicology studies, which asks, is it safe? So pharmacological studies try to prove that the new drug binds with the intended drug target, proteins, genetic material, or whole cells that are contributing to the disease. These studies have to be done both in vitro or in lab conditions using biological samples extracted from animals, and ex vivo in the whole animal in close to natural conditions. IND-enabling toxicology studies, on the other hand, investigate the drug candidate's toxicity, which can cause undesirable effects. Toxicology studies need to look at genotoxicity, or the drug's effect on genetic material. For example, does the drug being developed for cholesterol cause a gene mutation that can affect kidney function? And you also have to study the drug's toxicity on critical organ system, like what effect it has on the respiratory or reproductive systems. Pharmacology and toxicology IND enabling studies, making sure a drug candidate is safe to be tested on humans. Testing a new drug on humans or clinical trials is the longest and most expensive part of the drug development process. Clinical trials usually occur in three separate phases, phase 1 through 3. Proving a drug's safety and efficacy in clinical trials is a requirement for getting regulatory approval to manufacture and market the drug to the general public. Clinical trials can only begin after successfully completing preclinical trials, where the drug is tested on lab animals, and after getting an early stage approval from regulators to try it on humans. The three phases of clinical trials try to answer three basic questions. In phase 1, the question is it safe? Phase 2 answers the question, does it work? And finally, in phase 3, you ask, is the new drug better than what's already out there to treat the disease indication? Or is it better than the current standard of care? Completing phases 1 through 3 successfully leads to regulatory approval to launch the drug. Post-market data collection from real patients is sometimes referred to as phase 4 of clinical trials. Let's look at phases 1 through 3 in detail. In phase 1, the drug is administered to a small group of healthy volunteers, usually not more than 50. As discussed, the sole mandate of phase 1 trials is to see if the drug is safe. The concentration or dosage of the drug is gradually increased to check its safety, on genetic material and critical organ systems of the volunteers. The dose increase is stopped as soon as there are any signs of unsafe reactions. This kind of dose range studies forms the core of phase 1 trials. In phase 2, the drug is administered to a larger group, usually volunteers who have the disease indication. So if you're testing a new insulin variant, phase 2 volunteers would be diabetes patients. And the aim of phase 2 trials is to see if the safe dosage of the drug determined in phase 1 is effective. That is, it works to alleviate the condition. In phase 3, the drug is administered to many more patients, frequently in the thousands, with the aim of determining if it's markedly better than the current standard of care. There are two central tenets of phase 3 trials. The patients are split into random groups to minimize grouping based on genetic makeup, gender, or race. And the trials are controlled, which means about half of the volunteers are given the drug and the other half are given a placebo, like water. The groups themselves and who gets the placebo versus the drug are randomly picked by a computer. The patients, doctors, and trial administrators have no idea who's gotten what until the trials have ended. The 3 phases of clinical trials establishes that the drug is safe, that it works, and that it's better than the current standard of care, making it ready for regulatory filing and approval.
A big part of pharmaceutical R&D focuses on how a drug interacts with your body. This field is known as pharmacology. Pharmacology has 2 main components. Pharmacokinetics, or PK studies, focus on what your body does to the drug once it gets in. And pharmacodynamics, or PD studies, focus on what the drug does to the body after it gets in. Another helpful metaphor, PK studies is analogous to studying computer hardware. The keyboard, screen, cooling fan, and IC chips. And PD studies is analogous to studying computer software, word processing, email, sound mixing, etc. So PK studies look at where all the drug goes to in the body, for what duration and concentration it's present, and how and when it is excreted out of the body. For example, let's take a look at the humble paracetamol. Although usually safe, paracetamol when taken in high doses can cause liver damage. And this has been confirmed through PK studies. Because we found paracetamol fragments in the liver beyond certain dosages. PK studies thus help us set the safe dosage for drugs to minimize side effects. PK studies are also key to understanding the drug's bioavailability. Bioavailability measures whether the drug is available for use at the right site, for the right duration, and in the right concentration. That's why millions of diabetes patients have to go through the pain of daily insulin injections instead of taking it orally. That's because PK studies tell us that oral insulin just doesn't have the required bioavailability to treat diabetes effectively. Pharmacodynamics, or PD studies on the other hand, looks at the mechanisms through which the drug works. That is, how it treats the disease or disorder. PD studies focus on whether the drug is binding to the correct target, like the right genetic material, protein or pathogen, and how it affects that target to treat the condition. PK and PD pharmacological studies, understanding the relationship between the drug and our body.
It's often said that generic drugs are equivalent to a corresponding innovator branded drug. But it's not easy making an exact copy of an existing drug. It's not like innovator drug companies are lining up to give out their secret recipes to competitors as soon as the patent expires. Instead, generic companies have to reverse engineer the drug, break it apart to come to the individual components through publicly available information like what's on the innovator drug label, and also through physiochemical testing. But just because you know all the ingredients in a dish doesn't mean you can make it like a chef. You also need to know how everything goes together. So generic companies have to develop a manufacturing process that combine all the individual parts into a whole that's equivalent to the innovator drug. And there are two types of equivalence between two drugs. The first type is equivalence observable outside the body. Like these two drugs, they have the same active compound, paracetamol, in the same concentration. They also have the same dosage form and route of administration since they are both oral tablets. When tested in the lab, they would show the same critical physical properties, like the disintegration and then dissolution in water. This kind of equivalence, visible outside the patient's body, is called pharmaceutical equivalence. Two drugs that are pharmaceutically equivalent can still have different shapes and different inactive ingredients, like color and flavoring. The second type of equivalence is what the drug does inside the body, in their bioavailabilities. That is the active compound in the two drugs available at the target site in the body, in the same concentrations and over the same time? If the answer is yes, the two drugs are said to be bioequivalent. And when two drugs are both pharmaceutically equivalent and bioequivalent, they are said to be therapeutically equivalent. And that's the criteria set by most regulatory bodies to approve a generic drug. Prove that it is therapeutically equivalent to a previously approved innovator branded drug. Unlike chemical or small molecule drugs that can be exactly deformulated to make generic versions, it's not easy to reverse engineer a biologic drug. This is because the underlying biomolecule, usually a protein with thousands of atoms, is very complex. At its core, a protein is a unique sequence of amino acids forming peptide chains. This sequence is called the primary structure of the protein. These peptide chains then interact with each other in unpredictable ways, creating alpha helix and beta sheet secondary structures of the protein. And finally, the large protein molecule undergoes physical changes, or folding in 3 dimensions in even more unpredictable ways, to create tertiary structures. Primary, secondary, and tertiary structures of a protein are not just cosmetic concerns. They also affect its function, like its therapeutic effect on a disease. There are challenges in reverse engineering a biologic drug that goes beyond guessing the structure of the underlying protein, like determining what modifications, like glycosylations, were done to the base protein to improve its therapeutic properties, what host cells were used to culture the protein, and what excipients were added for stability or shelf life. In fact, a company looking to reverse engineer a biologic drug has to come up with an entire characterization and purification routine for the protein-based drug. It's like shooting in the dark. Because of these challenges, reverse engineering will never yield an identical version of the original biologic reference listed drug, or RLD. Only a similar one, a biosimilar version. And regulators reviewing a biosimilar drug application are not looking for pharmaceutical equivalence where every component of the two drugs are identical. Instead, they are looking to assess biosimilarity with the RLD. Assessing biosimilarity is a long drawn out and subjective process which involves as much statistics as biology. For this reason, getting an approval from regulatory agencies for a biosimilar drug can take up to 10 years, much longer than the typical 3 or 4 years taken for chemical generics. The bulk of the 10 years is taken up by clinical trials to prove that the RLD and biosimilar drug interact with the body in the same way. Clinical trials monitor a host of parameters, like the two drugs' safety profiles, their bioavailabilities, and efficacy inside the body. With results from clinical trials, statistical comparison to prove biosimilarity between the RLD and the biosimilar is done in 3 tiers. In Tier 1, the mean or average values of the parameters for the RLD and the biosimilar are compared. Having similar means for these values imply that the drugs are similar at a high impact level. In Tier 2, the standard deviation of the parameters for the two drugs are compared. This adjusts for statistical quirks, like pure chance or limited sample sizes. Similar standard deviations means that the two drugs are similar at a moderate impact level. And finally, in Tier 3, raw data from pharmacological and toxicological studies for the two drugs are compared. Tier 3 biosimilarity would only prove equivalence at very low impact levels. Considering the promise of biosimilar drugs in bringing affordable innovation to the masses, biosimilarity assessments is going to play a crucial role in the future of pharmaceuticals. A key part of drug development is the ability to conduct in vitro studies, that is, extracting living cells from organisms and growing and studying them in the lab. Cells extracted for conducting in vitro studies directly from a microorganism, lab animal, or human volunteers are called primary cells. Primary cells then go through a process called cell culture, where they are grown and multiplied in the lab. Cell culture requires maintaining ideal ambient conditions, like the perfect pH and temperature, and it also requires a culture medium to place the primary cells in, like this red liquid medium shown here. Culture mediums not only contain nutrients required for cell growth and multiplication, but also growth hormones and enzymes which can trigger or fasten growth. At perfect conditions, cells start multiplying in the culture medium. Not all cells are equal when it comes to culture. Fast multiplying cells, such as cancer cells and embryonic cells, are more viable than others. Once in culture and perfect ambient conditions, a primary cell starts to multiply, creating what is known as a cell line. A cell may undergo slight mutations at each step, but all cells in a cell line are descended from the same primary cell and has the same genetic information. In most cases, this is a finite process. A cell usually multiplies a maximum of around 50 times before it reaches a stage called senescence, beyond which it may be alive but does not multiply further. But in some cases, and for reasons not yet fully understood, senescence doesn't hit. Cell multiplication continues infinitely as long as ambient conditions and culture medium are maintained, creating an immortalized or never-ending cell line, like the cell line derived from Henrietta Lacks, an African-American mother of 5 who passed away from cancer in 1952. The primary cells derived from Henrietta's cancer gave rise to the HeLa cell line, named after her. Since then, the HeLa cell line has been crucial in drug development around the world, facilitating in vitro testing for many successful drugs like the polio and HPV vaccines and several cancer medications. For ease of use and distribution, large quantities of commercially available cell lines are cryogenically frozen in liquid nitrogen, forming a master cell bank or MCB. Researchers usually take out small portions from the MCB to conduct experiments. This subset is called a working cell bank or WCB. For a pharmaceutical researcher, choosing the right cell line for in vitro testing from hundreds of commercially available ones is crucial. They consider a variety of factors before choosing a cell line, like which species was the line extracted from, whether the cells in the cell line have the correct genetic marker or mutation required to test the drug, and whether the line is free from microbial contamination or mixing with other cell lines. In vitro studies in artificially grown cell lines, the bedrock of all drug development research. Daljit is excited about a new cancer drug in his pharma company's pipeline. His scientific and operations teams are ready to take it through the various stages of the drug development lifecycle, from discovering the drug compound to final packaging and market launch. Throughout this journey, Daljit's teams are going to be using hundreds, even thousands of biochemical, physical, and statistical analytical methods to guide them to a safe and effective finished drug. During the discovery phase, biochemical methods called assays are usually used to confirm the drug compound is binding with the correct cancer cells. During preclinical testing on animals, biological methods will be used to prove that the drug is safe to be tested on humans. In clinical trials, statistical methods will be used to prove that the drug is doing a better job of fighting cancer than a placebo. In the manufacturing stage, as a final drug is formulated, there'll be biophysical methods like spectroscopy to test the concentration of the drug compound and impurities, and to make sure that the drug stays safe and potent throughout. For Daljit and team, these analytical methods act like navigation tools in their drug development journey, telling them where they're going, what milestones to look for, and when they have taken a wrong turn. As important as developing analytical methods, validating them. Regulatory agencies require that data submitted with new drug application include results only from analytical methods that have been validated. A validated method meets 4 criteria. 1, it gives specific results. For example, an analytical method may show that the drug compound being developed for cancer is binding with cancer cells. But a validated method will also show that the drug compound is not binding with other random cells, giving a pertinent and specific result. 2, a validated method is robust, which means the method is rugged enough to still work even when there are small changes in ambient parameters, like the temperature in the lab or how steady the hand of a machine operator is. 3, a validated method is repeatable, which means if you run the method again and again, it gives the same results over and over. And finally, a validated method gives reproducible results, even when performed by different technicians in different labs at different times. Because of the large number of analytical methods involved in a typical modern drug development exercise, developing and validating the methods happen iteratively in real time. This means a method used in preclinical trials may still be in the process of being validated while the drug is already in the formulation stage of manufacturing. The aim is to be ready with the results from all validated methods in time for various regulatory submissions. So, one of Daljit's primary concerns is making sure his team follows Good Documentation Practices, or GDP. In a GDP environment, every single result, including failed results from all validated analytical methods, are catalogued and ready for review. With the method development and validation roadmap, Daljit feels confident that the new drug will be developed in a timely and efficient manner. Technology and knowledge transfer is all around us. Like when you teach your daughter how to bake a cake. You share a recipe, show her how to do it once, observe as she takes over, and then finally taste and evaluate the results of the knowledge transfer. Analytical method transfer in the pharmaceutical industry labs works much the same way. It happens between a pharmaceutical company and a contract research organization, or even between labs in the same organization. Basically, method transfer is when one party, a transferring lab, develops an analytical method or protocol, and another party, a receiving lab, needs to use it. In the pharmaceutical business, method transfer happens in 6 broad steps. In step 1, you make sure the receiving party can handle the method, based on their experience and skill level, and also the complexity of the product and procedures. Step 2 is document sharing on the method goals, equipment to be utilized, and standard procedures. Step 3 is working out a transfer protocol, which details the degree and type of analysis required, how to evaluate data generated from such analyses, and what the end result or acceptance criteria for the output should be. In step 4, you transfer the physical samples to be analyzed from the transferring to the receiving lab. Step 5 is training the personnel at the receiving lab. This can be done remotely or on site at the transferor or receiver facilities. And finally, in step 6, the receiving lab begins executing the method in close coordination with the transferring lab. The results are then shared, or in some cases, compared with results of simultaneous experiments carried out at both sites. If the results comply with the standards set out in the protocol, the receiving lab is ready to execute the methods independently. Failure to get compliant results would trigger an investigation to get to the root cause of the discrepancy, and a corrective and preventive action, or CAPA, is put in place to remedy it. A CAPA can include retraining personnel or tweaking the method itself, like by adjusting the acceptance criteria. Analytical method transfer, forming the basis of collaboration in the pharmaceutical industry. Medicines come in all types of dosage forms, solids, liquids, or gases that can be administered into the body through many different routes. From an ointment applied on the skin, to a tablet or capsule taken by mouth, to vaccines and biological drugs taken by injection, and even a gas or mist to be breathed in through an inhaler. And less common forms like vaginal or anal suppositories and medicines placed in your gums and cheeks. What route of administration a medicine takes depends on a host of factors, like the stability of the active ingredient, how local to the target area the specific application should be, and even the state of the patient. For example, there's no use prescribing an oral tablet to a patient who has trouble swallowing. Certain medications will not survive the rigors of the digestive tract when taken orally. A good example is protein-based biologic drugs. Our digestive system would just consider them food and digest it, preventing the drug from reaching the target site in the body. For quick action bypassing the digestive tract, injectable drugs are a great alternative. To understand how an injectable drug works, let's take a look at what lies beneath the skin. The top layer of the skin we touch and feel is called the epidermis. The layer below is the dermis, which has veins running through them carrying blood. Below the dermis is the hypodermis, also called subcutaneous tissue. This is mostly fat to regulate body temperature, but also has a limited amount of blood vessels. And below it all are muscle tissues, full of large blood vessels connected to the rest of the body. Intradermal injections that only penetrate the dermis are given by holding the needle almost parallel to the surface of the skin. These are rare and mostly done to do certain types of allergy and tuberculosis tests. Injections into the dermis, where the needle is held at a slight angle to the skin, are usually done directly into a vein running through it for intravenous therapy. Intravenous therapies are a great way to give the body nutrients when the patient is not able to eat or drink, and to fix electrolyte imbalances in the body. Subcutaneous injections, where the needle is held at a 45-degree angle, gets the drug into the limited vessels in the subcutaneous tissues. The medicines take time to distribute over the body because of these limited blood vessels. However, these injections are much easier to administer compared to intramuscular or even intravenous forms, and can be done by semi-skilled staff. Subcutaneous is how most biological drugs, from insulin to monoclonal antibodies, are administered. Finally, intramuscular injections, where the needle is held at a 90-degree angle, are done to get the medicine directly into the large and numerous blood vessels within the muscle. Intramuscular injections are a great way to get the drug into the body quickly and is widely used for vaccines and other drugs. It is less invasive and usually faster than intravenous administration. Oral dosage forms are the most common and least expensive way to take medicines. Oral dosages can be tablets or capsules and can take multiple forms even for the same medication. For example, paracetamol can be found as tablets and capsules to be swallowed whole, syrups that you can drink, or even chewable tablets, all taken through the mouth. There are two main criteria which makes a drug compound suitable for oral administration. One, it should be stable enough to survive the assault of the gastric juices in the stomach and small intestine without causing severe gastric irritation. And two, the drug molecule should be small enough to cross the cells that form the protective case of the intestine, or intestinal epithelium. Because of this, only small molecule chemical drugs are suitable for oral dosages. Biopharmaceutical drugs like insulin, vaccines, which are basically proteins, would likely get digested in the stomach. And even if it's not, would find it hard to cross the intestinal epithelium into the bloodstream thanks to their large size. Oral dosages are usually denoted on their packaging by PO, from the Latin per os, or by mouth. We typically think of topical dosages as ointments, creams, and lotions you apply on the skin. But topical actually encompasses all medications applied locally, directly at the location where you need the medication to work. So, topical medications could be an antibacterial cream that you apply on a bruise, but also eye or ear drops for a localized infection, or even nasal saline to clear a blocked nose. Topical dosage forms have some big advantages. They can be mass produced cheaply and stored for long times in room temperature. And they usually have limited and short lasting side effects compared to other routes of administration, usually limited to skin or eye irritation right after application. The drawback of topical forms is that they have limited applications, only on local ailments on parts that you can touch and feel. The vast majority of topical formulations are for application on the skin, an active compound held inside a base of cream, ointment, or lotion. For these formulations, the base is almost as important as the active ingredient itself. For example, some medications like topical steroids can gain strength by the order of 2 or 4 when it's based in an ointment instead of a cream. Topical dosage forms: efficient and affordable medications for localized relief. Think about the leading COVID vaccine candidates. There's one by Pfizer and BioNTech, and a second one by AstraZeneca and Oxford University. Both partnerships involve a big pharma company, in this case Pfizer and Astra, teaming up with a private or academic research lab whose strength lies in drug discovery. Now let's take a look at Crocin, GSK's easily available branded paracetamol. The GSK name appears in the front of the packaging, but turn it around and it says that Crocin is actually manufactured in India by Remidex Pharma of Bengaluru, a contract manufacturer for GSK. From drug discovery through R&D, clinical trials, and manufacturing, the pharmaceutical ecosystem involves various partnerships just like the ones we just saw, forming the pharma value chain. Let's explore the value chain a little deeper by looking at which players get involved at various stages of a drug's life cycle. As we've seen before, drug discovery involves a lot of scientific heavy lifting, so it's usually done by biotech or chemistry startups, academic labs, or niche research groups focusing on scientific research. In fact, for a lot of these groups, the explicit aim is to get a drug candidate to the proof of concept stage, with the hope that it'll then become an acquisition target for a larger pharmaceutical or biotech company. When it comes to routine development work and clinical trials, the pharma value chain is dominated by contract research organizations, or CRO. CROs perform routine but necessary work, such as figuring out ideal formulations for the drug candidate, establishing analytical methods, and even conducting preclinical and clinical trials. CRO leaders like Paraxel and India's Syngene use their experience, low cost structure, and a large pool of permanent scientific staff to get a new drug to market faster. Pharmaceutical manufacturing is frequently outsourced to contract manufacturing organizations, or CMOs. CMOs have huge supplier networks and are usually located in low-cost geographies with easy access to a skilled manufacturing workforce. And because they do manufacturing for multiple companies, CMOs can bring economies of scale to manufacturing processes. Because these reasons play to India's advantage, contract manufacturing is a $9 billion industry. Think about Think about the leading COVID vaccine candidates. There's one by Pfizer and BioNTech, and a second one by AstraZeneca and Oxford University. Both partnerships involve a big pharma company, in this case Pfizer and Astra, teaming up with a private or academic research lab whose strength lies in drug discovery. Now let's take a look at Crocin, GSK's easily available branded paracetamol. The GSK name appears in the front of the packaging, but turn it around and it says that Crocin is actually manufactured in India by Remidex Pharma of Bengaluru, a contract manufacturer for GSK. From drug discovery through R&D, clinical trials, and manufacturing, the pharmaceutical ecosystem involves various partnerships just like the ones we just saw, forming the pharma value chain. Let's explore the value chain a little deeper by looking at which players get involved at various stages of a drug's life cycle. As we've seen before, drug discovery involves a lot of scientific heavy lifting, so it's usually done by biotech or chemistry startups, academic labs, or niche research groups focusing on scientific research. In fact, for a lot of these groups, the explicit aim is to get a drug candidate to the proof of concept stage, with the hope that it will then become an acquisition target for a larger pharmaceutical or biotech company. When it comes to routine development work and clinical trials, the pharma value chain is dominated by contract research organizations, or CRO. CROs perform routine but necessary work, such as figuring out ideal formulations for the drug candidate, establishing analytical methods, and even conducting preclinical and clinical trials. CRO leaders like Parexel and India's Syngene use their experience, low cost structure, and a large pool of permanent scientific staff to get a new drug to market faster. Pharmaceutical manufacturing is frequently outsourced to contract manufacturing organizations, or CMOs. CMOs have huge supplier networks and are usually located in low-cost geographies with easy access to a skilled manufacturing workforce. And because they do manufacturing for multiple companies, CMOs can bring economies of scale to manufacturing processes. Because these reasons play to India's advantage, contract manufacturing is a $9 billion industry in India and is rapidly growing, driven by the manufacturing of injectable drugs. In fact, Divis Pharma, which is one of India's largest pharma companies by market cap, does not even make any brand name drugs or generics at all. Rather, they act as CMOs supplying active pharmaceutical ingredients to other pharma majors. From startups to CMOs, all participants in the pharma value chain work together to expedite delivery and bring down costs.